Here is my model of DNA replication. As you can see, I've already written out the template strand that was supplied in the directions. So the first thing that I'm going to do is I'm going to make the complementary strand. So what do I mean? DNA comes in two strands. So if this is the first strand, and these letters represent nucleotides, so this is thymine, this is adenine, this is cytosine, guanine. So there's always another strand running the other direction. So it's going to go 3' to 5', and we're not going to go into a whole lot of detail about what 3' and 5' means, but think of it as the direction that things are running on the freeway. And replication always goes from 5' to 3', so I'm going to start over here. And what goes with T, A. And what goes with A, T. And what goes with C, G. So I'm going to go ahead and fill all of this in. And please excuse my handwriting. Whoops, I put the wrong thing. Alright, so this is my complementary strand, and I'm going to use black for my labels. So this is my, what will be my template strand when I go to transcription and translation, and this is the complementary strand. Okay, so now I am a prokaryotic cell that is getting ready to divide, and so I need a copy of my DNA for each new cell. So I'm going to make a copy of this. So the first thing that's going to happen is, at the origin of replication, we'll say it's right here, I'm going to have an enzyme set down that is going to break the bonds. And I didn't draw the bonds in, so let's go ahead and draw them here. Okay, so think of these nucleotides as holding hands. And so this enzyme is going to come in, and it's going to break these hydrogen bonds and open up this double strand. And this enzyme is helicase. So you can think of helicase as breaking the double helix. Because if this were to get too much longer, this strand would look like this. So helicase is going to unwind that helix, and it's going to break the double bonds. So once helicase does that, and I'm going to go ahead and pause the video and go ahead and draw this in so you don't spend a whole lot of time watching, but it's going to break these two bonds, and I'm going to draw these in with the bonds separated. So helicase has broken the bonds between these nucleotides in the DNA, and we've opened it up, and we've got kind of a bubble here. And we're going to say that the helicase is going this direction. Now, the next enzyme to come in is called primase. Now, primase is an RNA polymerase, or an enzyme that makes a polymer, a big long string of RNA. Now, the next enzyme that's going to come in is DNA polymerase, but it can't bind to a single strand. So primase has to come in and make a primer. So it also has to go from 5' to 3'. So this is going to be the 5' end on this side, and it's going to be the 3' end here, because remember that we've got to go opposite directions, just like a divided freeway. So we're going to make a primer on this side. So we're going to go A. Now, you would think that T would go here to go with A on my tack here, but in RNA we don't have T, we have U instead, and G. Okay, so on this side we have to go the other direction, so we've got U, A, C. So now primase falls off, and DNA polymerase comes in and it can start making a polymer of DNA using these strands to make the right copy. So we're going to go this direction here, and we're going to go this direction here because we're going from 5' to 3'. So we're going to make a T here, because remember, this is DNA. So we've got T here. We can put an A and a G and a G and a T and an A. And as helicase moves along, then the DNA polymerase, and I'm going to put DNA polymerase 3, because it was the third one discovered, it's going to keep going as the helicase opens this up. So it's going to continuously replicate the strand in that direction. Now, DNA polymerase on this side, so we're going to go C, T, A, G, whoops, it should be C, excuse me, C, A, T, but all of a sudden it's run into a spot that hasn't been unwound, so it has to stop. Now, as the helicase opens up, primase is going to come over here and make another primer, and it's going to go that direction, and it's going to run into that primer. Meanwhile, down on this strand, as the helicase opens up the DNA, DNA polymerase 3 can keep going. So we call this strand here, this one here, and that yellow is not showing up well at all, is the leading strand, because it's going continuously. This one here is the lagging strand, because it's got to be replicated in these short stretches of DNA that we call Okazaki fragments. So here's my lagging strand, here's my leading strand. Eventually I'm going to get two strands of DNA that are going to be replicated, but I've still got these primers. So I'm going to pause and I'm going to erase some of this down here, and I'm going to do the two strands that are replicated. So DNA polymerase 3 has come through and replicated the DNA, and each of the nucleic acids match with the base next to them. Now, we've still got these RNA primers. We don't want those RNA primers there because RNA isn't as stable as DNA. Think of it as having a shorter expiration date. It's going to go bad, taste sour, within a couple of weeks, whereas DNA will last years. So, what we're going to do is we're going to take another enzyme, and we're going to take DNA polymerase 1, and we're going to come through and it's going to replace these primers with DNA. So it's going to come through and it's basically going to remove those RNA, and we're going to replace it with DNA. And now it's going to come over here on this side and do the same thing. We've got more primers on the lagging strand, so we're going from 5' to 3', so we're going to put a T in there, and we're going to put another T and an A. Now we got to get rid of this primer. Now, we've got to hook these strands together, but we've got to hook them in the 3' to 5' direction, and DNA polymerase can't do that. So we bring in another enzyme, and this one is called DNA ligase. And it's going to go through and it's going to put in the last of our little hookups in. So that's DNA replication. So, for extra credit, where in the cell is this happening? In prokaryotes, this takes place in the nucleoid region. That's where all the DNA is.